So let us proceed with today's topic, that how does a chromosome look like. So let us now have a quick look at what are the learning outcomes of this lesson. So at the end of this lesson, we will know how does a chromosome look like, or why a chromosome is acting as a carrier of heredity. Now the structure of chromosome, how does it look like? How we can differentiate between a sex chromosome and autosomes, and what are the different functions of chromosomes. So we will be discussing on this 4 important topics in this lesson. So let us see what is there in store for us in the first slide. Now let us start with what are chromosomes. So what are these? These are a strand of DNA molecules associated with proteins. So it is clear, right? They are DNA molecules associated with proteins. They are mainly found in the nucleus of the cell. Now see the red pointer here. This is a chromosome. Now this is mainly found in the nucleus of a cell. It is having different parts that we will discuss in the next all slides. But let us concentrate here. It is found in the nucleus of the cell. It is a hereditary vehicle which is containing genes. So what are these genes? Genes are composed of DNA and proteins. Now again, what is this DNA? This is a new term. DNA is a deoxyribonucleic acid which acts as a genetic material. And this is the chemical basis of heredity. So what is the chemical basis of heredity? DNA or deoxyribonucleic acid is the genetic basis of heredity. Now what is a gene? Now gene is a segment of DNA that codes for the synthesis of a specific protein. So it is a segment of DNA that is coding for the synthesis of a specific protein which controls the expression of a particular characteristic in an individual. So the gene is always called the basic unit of heredity which is found on a chromosome. So this is DNA. This is the deoxyribonucleic acid. This is a chromosome which is having different parts. Now the different parts of the DNA molecule we will study in the later slides. Now let us be clear about what is a chromosome. Now the genes which are the main carriers of information from one generation to another, or which carries the genetic material. So that is the deoxyribonucleic acid, which is the chemical basis of heredity. Now let us now move on to the next part, which will discuss about the chromosomes in detail. Now let us start here just with the diagram. How does a chromosome look like? So these are the structures of chromosome. So chromosomes greatly they can differ in shape and size. You can see here are 4 chromosomes which all are differing in different size. Okay, their shape is also different. Now the chromosomes may differ in the position of the centromere. Where is the centromere? This is the centromere. Now if you remember the previous cell division slides, I think you should remember where is the centromere. So if you are not able to recall, please go through them. I am providing the link each and every in almost many slides. I am providing the link upwards. So go to those slides and see that where is the centromere located. So this is the centromere. Now here we are seeing a new term called kinetochore. So what is a kinetochore? This is a protein which is present near to the centromere, and if you remember the formation of spindle fibers in all the cell divisions, these proteins are binding those spindle fibers. So these kinetochore, they have a role in cell division. They are present near the centromere. Now let us discuss what is a centromere. Now this is a point on the chromosome where the spindle fibers are attached. Now this is a point where during cell division the spindle fibers were attached. And to what they were attached? They were attached to this kinetochore protein. Now let us move on to these diagrams. Now if the centromere is near the middle of the chromosome is called metacentric. And if the centromere moves towards the end, or it is towards one end away from the center, it is called acrocentric. So this is a metacentric chromosome. Next we will see that it is moving away from the center or towards one end. It is acrocentric. And if the centromere is located at the end of the chromosome, what is it called? It is called telocentric. And when it is not in the exact middle position, either towards the top or towards the down, it is called submetacentric. So is it clear for everyone? Please make it clear. If you are not clear, if you are having any doubts, please do ask me. Please do write in the comment box. I have been telling you the same thing, that please do write your query so that I can make it clear in the next video. And as it is a distant learning or online learning, I can't see your faces, or rather I can't see your feedback. So please do write your feedback, whether you are liking it, whether you are able to understand or not. So let us do a quick revision again. Centromere in the middle, metacentric. Near any one end is acrocentric, that is moving away from the center. At the end of the chromosome, it is telocentric. It is a little bit away from the middle part, centromere. It is called submetacentric. Now kinetochore is the protein present near the centromere, which helps in the binding of the spindle fibers during cell division. Now chromosomes are formed by the two sister chromatids. You can see these two are sister chromatids. So these two sister chromatids combine to form a chromosome. Now each chromosome at the start of cell division having two chromatids. They are joined at this point of centromere. But after we have seen that spindle fibers are formed, at a time the spindle fibers, they contract and they help in the separation of this sister chromatids, and these two move towards the opposite poles. I think you remember that. Spindle fibers are attached to this place and they are being pulled towards the opposite poles. So this is, and on completion of this cell division again, the chromatids are called, the sister chromatids again are called, these two combine to form again chromosomes. So these chromatids then again condense and form the very thin thread-like structures that we can call the chromatin fibers. Okay, is it clear for everyone? They again condense after the cell division. These chromatids again condense and form very thin thread-like structures called chromatin fibers. Now these chromatin fibers, what they are made up of? They are made up of DNA and histones with very small amount of RNA. So as of now, we have made this thing clear. So let us move on to the next slide. Now here comes chromatin. So what is it? What is a chromatin? We have told that during cell division and all, we have seen chromatin fibers. So what is it? So this is a chromatin fiber. This is a chromatin fiber. So it is consisting of two strands of DNA and proteins which are mainly histones. So how does it look like? It looks like beads on a string. So DNA along with histones forms this chromatin fibers. And this DNA is almost 40% and histone proteins are almost 60%, which form the overall part of a chromosome. Now here we can say that DNA is negatively charged and histones are positively charged. So we know that positive and negative charges attract each other. So how does this DNA histone complex look like? They look like beads on a string. And this DNA histone complex is called chromatin. This is the chromatin fiber. So a chromatin fiber is made up of DNA as well as histone proteins, where the histone proteins are positively charged and DNA is negatively charged along its length. See, how does it look like? When we are seeing it in close up, it looks like beads on a string. So the string is DNA and these are all histone proteins. So DNA helix combines 8 histone molecules. Now you can see my pointer over here. This is the DNA which is combining 8 histone molecules to form a nucleosome. So what is a nucleosome? The DNA is wound on nucleosomes. And it is looking as a beads on a string. And there are 2 million nucleosomes among the 46 chromosomes. So what are these 46 chromosomes? Our human body consists of 46 chromosomes, of which 22 pairs are autosomes and the 23rd pair is the sex chromosome. Okay, is it clear? 23 pairs of chromosomes are there. That is 46 chromosomes are there, of which 22 are autosomes and the 23rd pair is the sex chromosome. So is it clear for everyone? So chromatin, what is it? It is the DNA histone complex. The DNA is a negatively charged thing along its length and histones are positively charged thing. So the DNA helix combines with groups of 8 histone molecules to form this nucleosome. And how does it look like? It looks like beads on a string. And human body consists of 2 million nucleosomes. So I think till this it is clear to you all. So please go through your textbook. Please go through my videos if you want to revise and recall the previous cell division episodes. So I'll put the link, and please don't forget to subscribe to my channel if you are liking my teaching and my videos obviously, and don't forget to like and share my videos. Now let us move on to the next slide. Now here let us start with the most important slide, which is the structure of a DNA molecule. Now when you are studying the structure of chromosomes, you should be very clear with all the basics. If your basics are not clear, when you are moving to higher classes, you can't if you can't remember these basic models, you won't be able to understand the replication, transcription or translation processes. So those in higher classes you will be studying. So now for the basics, you need to make it very clear. If you are not thorough with this, it will be difficult. So when you are studying this, you go through your textbook, you go through my slides again and again to make the concepts clear. Now let us start with the structure of a DNA molecule. Now first, Rosalind Franklin, a person in the 1953, he studied the shape of a DNA. But he didn't work out on the structure of the DNA molecule. This was found out by Watson and Crick in 1953. He told that it is a macromolecule, that is, it is a very large molecule having 2 polynucleotide strands. We can see one here, another here. 2 polynucleotide strands which are complementary in nature and wound around each other in a double helix. You can see the structure. It is a double helical structure. So in one word, we can say that DNA is a double helix. Okay, is it clear? Now, it is made up of polynucleotide strands. Now, these strands are anti-parallel. They are composed of repeated nucleotides. Now, what are these nucleotides made up of? These nucleotides are made up of a phosphate, a sugar, and a nitrogenous base. So these polynucleotides are a phosphate, a sugar, and a nitrogenous base. So this polynucleotide strand is actually a sugar phosphate backbone. So here we can see that it is a sugar phosphate backbone which has nitrogenous bases arranged at right angles, giving it a ladder-like arrangement. If you see it, you can feel that we can climb the ladders. So this nitrogenous bases are arranged at right angles, giving it a ladder-like arrangement. So what are these nitrogenous bases? There are 2 purine bases and 2 pyrimidine bases. So what are those? Those are guanine, cytosine, adenine and thymine. These 4 purine and pyrimidine bases form this nitrogenous bases ladder-like arrangement. So we have given it different colors. You can see that if this is adenine, this is thymine, this will be guanine and this will be cytosine. So in one word we can say that it is ATGC base pairing. So remember this term, this is ATGC base pairing. It is having 2 purines and 2 pyrimidine bases. And learn this spellings also properly so that you don't make a mistake in purine and pyrimidine bases. So as you are all learning chemistry, you know what are nitrogenous bases. You should know also this purine and pyrimidine bases and how they are forming this ladder-like structure. So is it clear for everyone? Let us move on with the next slide. Now in this slide we will see quickly the difference between a sex chromosome and autosomes. Now in this diagram what we can see is any human karyotype. So there are, we can see that 22 pairs of autosomes, and this 23rd pair is only the sex chromosome or allosomes. So autosomes, these are the genes which control all the somatic traits, and they don't play any role in sex determination. That it play roles, it play role in determining the physical characteristics, the skin color, the hair color, the eye formation, everything. So these are all related to the somatic cells, that is the body cells, but they don't determine the sex of a person. So this 23rd pair is the sex chromosome which will play a role in sex determination. So 2 members of each pair of a homologous autosome are similar in shape. We can see they are almost similar in shape, but it is not true for the sex chromosome. Here you can see that this X is much larger than Y. Y is much smaller. But in all these autosomes we can see that mostly all the pairs are similar in shape, almost similar in size. Not in shape exactly, but they are almost similar in size. And there is a huge difference in the size in this sex chromosomes. So there are 22 pairs of we can say the 22 pairs of autosomal chromosomes or autosomes, and the 23rd pair is the sex chromosome, or it is called the allosome. So let us move on to the next slide. Now, though this part is not there in your portions or syllabus, let us move on to this quick view of how does a male and a female chromosome look like. Now you can see that male chromosome is X and Y. Here the X is much larger than Y. Okay, so it is the male chromosome, and here female, you can see both the X's are almost similar. So these are similar. So the sex chromosome of human female is called homomorphic, and that of a human male is called heteromorphic. Now females produce one type of gametes and are called homogametic. So females can produce only X chromosome, so they are homogametic, whereas the males can produce 2 types of gametes, that is X and Y. So they are called heterogametic. So this is heterogametic chromosome, and this is homogametic. So a human male is heteromorphic as well as heterogametic, whereas a human female produces, they are called homomorphic and homogametic. So these are some new terms you should remember. So this is not so important, but just for your knowledge, you should remember this, so that higher classes it will help you This all 4 terms are a little bit new, and they are all related to the structure and functions of chromosomes. So for that I made it clear. Now only the functions of chromosome is left. Now here just a quick overview of the functions of the chromosomes. So we have told in the first slide itself that they are acting as hereditary vehicles. So they are carrying the genetic information from one generation to the next. So they are acting as genetic containing hereditary vehicles, and they contain genes. Again, the next one is they control the synthesis of structural proteins, thus help in cell division, cell repair and growth. So they play a very important role in cell division, cell repair and cell growth. Whereas the third point, we can see that they by directing the synthesis of enzymatic proteins, chromosomes control the cell metabolism. So first one, acting as a carrier of hereditary vehicle, helping in cell division, cell repair, cell growth. By directing the synthesis of enzymatic proteins, they control the cell metabolism. Now chromosomes guide the development and control of cell differentiation. Now sex chromosomes, the last one, that is XX, XY and XXXO chromosomes, they determine the sex of individuals. So when you are studying genetics, next chapter heredity and genetics, when I'll be teaching you, then you will study all this in details. How these genetic traits are carried from one generation to another, how this XXXY chromosomes are acting. So we will study it in detail in the next videos. So as of now, this is the function of chromosomes. So this is the complete part of structure and function of chromosomes with all other topics which I have explained. So please do go through your textbook, whatever book you are referring. Please do study my previous videos on cell biology so that it is clear to you till now whatever I have taught you. And don't forget to comment and don't forget to subscribe to my channel so that whenever my next uploads are coming, you can get it first and you can study those lessons also. And please do like and share so that all your friends can get benefit from all my teachings. Thank you.